Okay, thank you. No disclosures. My lab is interested in looking at how brain networks develop over the lifespan and how that relates to cognitive development. And so what I'm going to talk about today is how this whole global signal regression ongoing debate affects people's analyses in this realm. When you're thinking about things like changes over time, when you're thinking about group differences, when you're thinking about case control analyses, how does everything we've heard about today affect your interpretations of some of these questions in lifespan, cognitive and neural development? And so in the early days of the pandemic, I was feeling philosophical and had a lot of time to sit around and think about this idea of spontaneous neural activity and what it means for us, especially in this field as we're kind of growing to think about these signals in the brain and how we interpret them, how we clean them, and how we separate signal from noise. What is the brain signal and what is the brain noise that we want to get rid of? And I think this is something that we think about a lot in resting state fMRI because we're not modeling a task and we're not seeing what goes along with it. We're looking at intrinsic functional organization of the brain, but in order to do that, we have to take care of all these other artifactual things that go along that are really hard to tease apart in many ways. And so I just want to point, if I can wax philosophical for a moment, about like sometimes in neuroscience, the things that we used to consider noise are now really we know to be signals. So spontaneous single unit and population activity measured at the single neuron level, we used to think that's just noise. Like, why do we care about spontaneous activity? But now, there's sort of decades of work showing the significance of this activity for neural function. And things like spontaneous large-scale network activity, similarly, we used to just kind of discard as not important, but now it's been shown over the years to be very critical for neural function. Variability in neuronal spiking has also in the past been kind of dismissed as something uninteresting, but is now thought to kind of increase the sort of regime of possible responses of neuronal populations. So it's really the variability itself of the spiking is something that we now think of as important for function and not just something to be washed out or averaged away. And bold signal variability is something we've looked at a lot in the lab recently, just how the signal varies over time. May not be, people used to not consider it super important, but now it's been shown to be related to things like cognitive flexibility and lots of interesting theories about why a variable signal might be good for letting us flexibly respond to environmental things. And so here we get to the global signal. Is it signal? Is it noise? I cannot believe we're still talking about this. It's just like the first paper I ever wrote using resting state fMRI data was looking at the default mode negative correlations in 2009. And there we did the analyses both with and without global signal regression because everyone asks you to do it both ways anyway. So, and that is still the case. So it really does seem to be, we can't let this go. And so I think what we just need to think about is what is the global signal, right? Is it signal, is it noise? Well, it's obviously a little of both, right? There's motion artifacts, scanner artifacts, all these things get cleaned up very nicely when you do global signal regression. But there's also neuronal activity, and that's been shown in monkey electrophysiology and a lot of other work that I don't have time to review today. And this is, again, just showing what Tom mentioned earlier, that you can see if you use things like complex principal components analysis, there's a large global signal pattern that comes out, and it's kind of a mix of standing and traveling wave activity. And I just wanted to show this video because it looks cool. But basically, if you think about treating the global signal as something that could potentially be of interest, you can do things. This actually was a project that started as a joke after the Singapore meeting in 2017, and sat around with a few colleagues and thought, like, wouldn't it be funny if we just took the global signal and tried to relate it to all these behavior and cognitive measures just the way you would with something like a functional connectivity matrix and try to relate, do brain behavior correlations. So we did that, and we just took this top is just showing you the average global signal in some HCP data. And we basically just kind of used principal components analysis here again to look at global signal principal components, and they do sort of recapitulate some of the patterns we see in terms of task positive, task negative networks, and things of that nature. But if you compute the global signal across, the way you've heard about it computed, and then make sort of a beta, correlate each voxel of the brain with the global signal to see the global signal topography and how that's expressed across the brain, you can create a global signal beta map, and then subject that to a canonical correlation using all of these variables that are in the HCP data. And we sort of recapitulated this positive negative axis of behavioral correlations that we saw in an earlier paper by Steve Smith, where that was a correlation that came from looking at functional connectivity matrices related to behavior. But basically, you can get almost the same exact brain behavior correlation that you saw in that paper just from using the global signal weights, which, in some cases you might have just discarded. It turns out all that same information is just also there in the global signal weights. So this is just suggesting that observable individual differences in the spatial topography of the global signal, you can see them. We've seen that in many studies, and that the global signal may contain a rich source of information related to these trait-level cognition and behaviors that we're always trying to do brain behavior correlations, and we're trying to clean up the data to find those correlations. But sometimes the thing that we're throwing away is one of the things that's most correlated with the behavior. And so I started out talking about the impact that this preprocessing step has on, if you're looking at group differences, age-related differences, clinically relevant questions. And so Jason Nomi in the lab basically took the Nathan Klein Institute data set, which includes resonance data, fMRI data from individuals between 6 and 85, so a nice, hundreds of subjects covering the lifespan. And if you break that down into, 10-year kind of bins, 6 to 15, 16 to 25, et cetera, you can see some very they're not very big changes, but there's differences in the global signal topography in these different age bins. So there is, at the surface level, a little bit of difference there you can see. But what's most interesting, I think, is if you look at the relationships of global signal topography as it changes over the lifespan. So now, instead of trying to predict behavior from the global signal, we're just looking at age as the variable of interest here. And maybe I'll just focus on the thalamus result because it's easy to see this sort of linear increase, which is showing that the weights, the global signal is weighted more in the thalamus, and that weight increases in older individuals compared with younger individuals. So they're sort of the sort of topography of the global signal, it changes across the lifespan in a way that there's sort of more of it in the thalamus, in older than in younger ages. And there's different relationships with different brain regions. So the putamen shows kind of the opposite effect with age. And you see another subcortical region here, the nucleus basilis, which shows a kind of U shape, or a different kind of trajectory related to age. So these are all things that are, I think, of interest if you're trying to make age-related or you're trying to explore age-related relationships in your data. And it gets very complex if you look at different brain regions. So there's the frontal eye field shows that, and some of the cortical regions show these U-shaped effects as well, quadratic effects over age. And so this leads us to, my least favorite thing to do is methods pipeline papers, but sometimes it's what you have to do in order to figure out what are these different pre-processing steps actually showing you. So this is, thankfully, Lyndon Parks and Alex Fornito did a great job of looking across 20 different pipelines, and some have global signal regression and some of them don't. And many of you have contributed to doing these wonderful pipeline papers that are a real pain, but everyone thanks you for them. But basically, they were looking at the influence of these different pre-processing steps on motion and other benchmarks of data quality. And basically, one nice thing was that a pipeline that just involves ICA aroma denoising was relatively successful based on all of these benchmarks. So that doesn't involve global signal regression at all if you want to just sidestep the issue. But the addition of GSR generally improves the performance of all pipelines across most metrics. So you sort of can't get away from this denoising benefit that you get from global signal regression. Too bad. So we're trying to do this now, comparing pipelines with and without global signal regression in this age, in the 6 to 85 sort of lifespan cohort. And the picture gets kind of complicated because, well, we're also trying here the temporal ICA method from Matt Glasser, but it turns out that not only does global signal regression affect the functional connectivity matrix differently across the lifespan, as you might have expected from what I showed you before, but also things like filtering, those steps also make a huge difference in these lifespan FC matrices. And so this is a paper that's just in progress now that Jason Nomi is working on, looking at how the functional connectivity matrices are affected by these kind of different pre-processing steps, some of which include GSR and some don't. And so, it's just kind of showing you like which of these pipelines are more correlated with each other. But I wanted to just kind of highlight the fact that the neurobiological information that's kind of contained within the global signal seems to be possibly differing across the lifespan, across development. And we sort of have to carefully consider whether or not to remove the signal when you're interested in age-related functional connectivity differences, because it just might be differently expressed in these different age groups, just like we've seen in previous work in schizophrenia and bipolar and autism. Sometimes in case control kinds of analyses, the global signal is expressed differently in these different groups, and so the question of whether or not to regress it out is the question of, do you want to differentially regress something from your different groups when you're making these kinds of comparisons? And so, so pre-processing, pipeline filtering, and global signal regression all have differential influences on age, functional connectivity association strength. So it's just something to consider. And as also we heard about earlier, these global signals are really linked to things like pupil diameter, heart rate, respiration, all kinds of physiological measures. They seem to be very tightly linked, and they kind of all go together. And so this may not be surprising. So this paper, I think we had a poster on this last year at OHBM, and the work is still under revision, but any day now. So this is collaborative work from Shella Kilholtz, Katie Chang, Nathan Sprang, and many others. We keep adding collaborators because we keep adding new data sets, because we keep finding new things to look at here. But the idea of we're not going to get a simple answer to this question about global signal regression, as you can probably guess from now. So this is just the idea that the brain-body synchrony can be captured in one spatiotemporal pattern across fMRI, EEG, and peripheral physiology signals. So the global signal really is integrated with all of these other sort of physiological measures. And again, I won't go into this because Dimitri did a nice job of going over this dual layer model of the global signal. But again, I think it just speaks to our inability to right now know how to separate the signal from noise when we look at this. We're still trying to figure out how much of it is something to be explored in terms of neural information, and how much of it is just the scanner artifact, the motion artifact, all of the other things we want to get rid of. So is global signal or noise? I don't know. I do like to write opinions about it, but the truth is we can't just say it's signal or it's noise, because there's so many different ways of trying to think about this question. And I remember Peter Bandettini wrote a nice brain blog about this a few years ago, where he wanted to say that, it shouldn't be removed, and we have a consensus on that. And I thought that was fun, because I guess we haven't reached a consensus, because here we are, again. So I think it's kind of fun to be humbled, actually, by how little little consensus we've come to, because it just means that there's a lot to be explored in this signal. And so I think personally, there was a nice paper a few years back from Murphy and Fox, where they talked about whether GSR is a useful pre-processing step likely depends on the specific question one seeks to address. Results must be interpreted properly within the context applied in different methods may provide complementary insights. And so I think that when you're looking at things like group differences, then it may be prudent to be more cautious in doing this type of regression for the reasons that I mentioned. And I think we don't have to decide. I think we can be friends, and I think we can use global signal regression when we need to for certain questions and other times not. I don't think we need to pick one pipeline at this point in time. I don't think it would be wise to anyway. So thanks to the collaborators, the organizers, and to all of you for keeping the debate alive. And I'll take questions now at the end.
So the question for you, Lucina, is like, if we imagine that we want to capture individual differences, is it possible that by regressing out the global signal, we also remove meaningful individual difference? So can we do group-level regression rather than individual-level regression to exclude the influence of global signal or uninterested signals?
Interesting question. Group-level regression of global signal? Well, if it's differentially presented in different individuals, then doing a group-level regression would differentially affect each individual. So I think that's always my problem, is like we're regressing a different thing from, well, I mean, maybe that's just how it goes, but I'm not going to take a stand on it. I'll leave it to the next speaker.
Thank you so much. Next one is